In 2025, a single DNA sample from a man living on a Montana reservation broke open a mystery that geneticists had quietly buried for nearly 3 decades. What they found inside his blood wasn't just unusual, it was a genetic switch, dormant under normal conditions, that only activates when the human body is moments from death, and it appears almost exclusively in people with type O blood. This discovery didn't make the evening news, but it may have just rewritten the story of how our species survived. Type O blood is everywhere. It's the most common blood type on Earth, carried by roughly 44% of the global population. Hospitals depend on it. Emergency rooms stockpile it. It's known as the universal donor because it can be safely transfused into anyone, regardless of their own blood type. Most people know this much. What most people don't know is what the global distribution of type O actually looks like, and why it makes no genetic sense. In parts of South America, up to 100% of certain indigenous communities carry type O exclusively. Not majority, not dominant, exclusively. Type A doesn't exist in these populations. Type B has vanished entirely. In a 1980s study of Native American communities in Arizona, 99% of participants tested type O. The global average is 44%. These populations are sitting at 99 to 100. Population geneticists have a term for when a trait spreads this completely: fixation. But fixation through random chance, genetic drift, takes thousands of generations and leaves traces of variation behind. Here, there are no traces. Other blood types weren't just reduced, they were erased. Something removed them. And new DNA evidence is finally revealing what that something was. For decades, science had a comfortable explanation for this. The story went like this. Roughly 15,000 to 20,000 years ago, during the last ice age, small bands of hunter-gatherers crossed a land bridge connecting Siberia to Alaska, a now submerged stretch of land called Beringia. These groups were tiny, maybe a few hundred people. If they happened to carry mostly type O blood, isolation would do the rest. Cut off from the world, the founding gene pool simply locked in. It was a clean theory, logical, widely accepted. Then ancient DNA research demolished it. When geneticists extracted and sequenced blood type alleles from skeletal remains of ancient Siberians, the very populations believed to have crossed into the Americas, they found something the old theory couldn't accommodate. These ancient Siberians were genetically diverse. Type A, Type B, and Type O were all present in significant numbers. Multiple blood types crossed that land bridge. The genetic record confirms it. So the question that should have ended the debate became impossible to ignore. If Type A and Type B entered the Americas, where did they go? They didn't drift away slowly, they didn't gradually thin out across generations. In the ancient skeletal record, researchers traced them moving south from Alaska, and then watched them disappear. Present in early Alaskan remains, fading in the Central Plains, gone entirely by Mesoamerica, absent without exception in early Andean populations. This wasn't drift. The pattern looked more like a purge. In 2024, a coalition of geneticists from Canada, Brazil, and South Korea published findings from the most comprehensive ancient DNA blood type study ever conducted. Sequencing genetic material from skeletal remains spanning from Alaska to Patagonia, they traced Type A and Type B alleles across thousands of years of prehistory. Their conclusion, buried in a single line of the study's abstract, was blunt. The data imply a directional selection event, potentially immunological in origin, favoring O alleles over all others. Not random drift, directional selection. Something in the ancient American environment had systematically eliminated Type A and Type B carriers, while Type O individuals survived. The most likely mechanism is pathogenic. Certain bacteria and viruses use blood group antigens as entry points into the body's cells. Type A and Type B cells display these molecular markers on their surface like open doors. Type O cells display neither. To pathogens that evolved to exploit A and B antigen specifically, Type O blood is effectively invisible. Supporting evidence appeared in the highlands of the Andes, where researchers studying ancient dental enamel found microscopic antibody traces locked inside the teeth of ancient individuals, immune records of battles fought against pathogens that no longer exist anywhere on Earth. The carriers of those antibodies were, in nearly every case, type O. The other blood types weren't just less common in these populations, they were absent from the archaeological record in the same layers where epidemic evidence appeared. Type O didn't spread because it was lucky. It spread because during at least one catastrophic disease event in prehistoric America, it was the only blood type that survived. Which brings us back to Montana and a man named Thomas Crawford. In early 2025, Crawford, a member of the Blackfeet Nation, participated in a large-scale ancestral mapping project. His results were expected to be routine. They were anything but. Crawford's DNA carried a rare deletion mutation in the ABO gene cluster, a section of genetic code missing entirely rather than simply altered. Researchers identified it as a variant found previously in only one other source, a 12,000-year-old skull recovered from permafrost near Lake Baikal in Siberia. The same lineage, separated by 12 millennia. The genetic fingerprint was identical. That alone would have been remarkable. But it was what appeared alongside this mutation that stopped the research team cold. In a region of Crawford's genome associated with immune system regulation, an area long dismissed as non-functional junk DNA, there was an unusual fragment of genetic code. Under normal conditions, it was completely silent. But when researchers ran advanced immune modeling simulations, exposing the sequence to scenarios of extreme biological stress, something unexpected happened. It switched on. Once active, the sequence appeared to coordinate a cascade of immune responses, stabilizing inflammation, protecting organ function, regulating the body's reaction to systemic infection. In the simulation, it behaved like an emergency survival protocol, written directly into the genome, one that only triggers when the body is already in crisis. This fragment appeared disproportionately in type O carriers, and according to internal records later surfaced by a researcher familiar with early genome project documentation, a sequence matching its description had been flagged during initial human genome project analysis in the late 1990s, then quietly removed from public datasets without published explanation. Whether that removal was administrative or deliberate, no one has formally confirmed. But the sequence is back in the scientific record now, and its implications are significant. Type O blood may not simply lack certain antigens, it may carry something the other blood types don't, a latent biological defense system, built through generations of survival pressure that activates only when everything else is failing. Crawford's case didn't exist in isolation. As researchers cross-referenced his genetic profile against global databases, a pattern emerged that stretched far beyond North America. In 2025, skeletal remains excavated on Easter Island, Rapa Nui, dating back roughly 900 years, revealed an overwhelming prevalence of type O blood. That alone wasn't surprising. What was surprising was a secondary genetic marker present in those same remains, an immune-modulating variant also found in high-altitude Andean populations on the South American mainland, thousands of miles away across open ocean. The same rare immune variant, two populations, separated by the Pacific. DNA evidence has previously confirmed that Polynesian voyagers made contact with South American peoples approximately 800 years ago, carried in the genetic record of living Polynesian islanders today. What this new evidence adds is a biological dimension to those voyages. The people who crossed that ocean weren't just trading sweet potatoes and bottle gourds. They may have been transmitting, unknowingly, the same genetic survival tools forged by thousands of years of epidemic pressure. From a frozen skull in Siberia, to ancient teeth in the Andes, to bones preserved in volcanic soil on Easter Island, to a DNA sample submitted by a living man in Montana, the same thread keeps appearing. Type O blood isn't simply common. It is, in the genetic record, the survivor. The last one standing after events so catastrophic they erased every other option. What science is now beginning to understand is that type O blood's prevalence in certain populations isn't a statistical accident or a quirk of geographic isolation. It's a scar left by ancient outbreaks that no longer exist, by pathogens extinct for thousands of years, by survival events so severe they restructured the genetic landscape of entire continents and left a hidden emergency protocol embedded in the human genome. The people who carry type O blood today are the descendants of every one of those survivors. Their blood carries the memory of those trials in its very structure. And as geneticists now turn their attention toward emerging pathogens, antibiotic-resistant infections, and biological threats still unknown, the question this research quietly raises is whether that ancient survival code still works. The answer, if Crawford's genome is any indication, may be yes. The genetic record has been quietly telling this story for thousands of years. We're only now learning how to read it. If you found this investigation compelling, subscribe to History Diary. We cover the breakthroughs in DNA archaeology and genetic history that are rewriting what we thought we knew about the human past. And if you have a blood type you're suddenly thinking differently about, drop it in the comments. The conversation down there is always worth reading.